Ever wonder why so many studies say red meat will kill you, but people on carnivore are thriving? Well, let's expose the hidden flaws in nutrition science that keep you stuck eating the wrong foods. Hey everyone, my name's Mark Bates. I'm a metabolic health coach, and this is part of my Roasting Sacred Cows and Nutrition series. Why most nutrition studies get it wrong about the carnivore diet. Okay, let's break this down. There's a big problem with epidemiological and rodent studies in determining human health, and these are by far the two most common studies you'll see in research that people will write about. Okay, first let's start with what is an epidemiological studies. It is a form of statistics, and it was originally built and designed to help track communicable diseases. In fact, one of the greatest scientific triumphs of epidemiological science was in London in Victorian times when they used this statistical science to track where cholera was coming from in the population, and they were actually able to isolate it down to one single water fountain. And this is where people had these communal water fountains where they everyone collected their water, they took it home and they used it to cook with and clean and on all that. And they found by correct use of this statistical science that there was one water fountain that was highly contaminated, and by shutting down that one fountain and getting people to get their water from a different source, they were able to drastically reduce the amount of cholera in the city, reducing deaths and sickness and all kinds of great things. Absolute wonderful use of this. Okay, the problem is when you're doing it for nutrition, there are lots of limitations on it. Okay, so while epidemiological studies statistically do have some validity, they can only provide clues about population level diet disease relationships, right? Uh, but they but when they're used as the primary evidence for what constitutes a human healthy diet, especially in nutrition policy, they have serious logical and methodological limitations. On the same basis, rodent studies are incredibly common in nutrition research because mice and rats are inexpensive, reproduce quickly, and can be housed in controlled environments. But when it comes to determining what works for human health and diet, they have serious translation problems. So let's look at these very quickly. 1. Correlation does not equal causation. And you hear that a lot, but let's make sure you understand it. Epidemiological studies, especially observational cohort studies, which make up the vast number of all studies published on nutrition, are identify associations, not causal links, right? Right? And the classic example of observational studies and causation and associations are on a bright sunny day, everyone's at the beach, and they all go to this ice cream parlor or ice cream stand, buy ice cream, and then at the end of the day they have sunburn. So obviously it's the ice cream causing the sunburn. It's a valid epidemiological observation and association. It's worthless, but it's a valid one. Okay, so if a study finds that people who eat more red meat have higher heart disease rates, it does not prove that red meat causes heart disease, right? Like people who eat ice cream ice cream does not cause us sunburn on people who go to the beach. Other factors, lifestyle, socioeconomic status, smoking, all that sort of stuff may be the real drivers, right? And it's hard to identify all those things. So, from a carnivore and ketogenic implication, red meat often appears unhealthy in such studies because meat eaters in modern populations tend to eat it alongside refined carbs. Burgers, which include a bun, fries, and soda, engage in more risk-taking behaviors, right? They smoke occasionally, right? That's becoming less, but many people still smoke. And they follow the standard American diet, right? not a clean carnivore diet regime. So you have all kinds of confounding factors. Okay, you have confounding variables, right? Confounders are other factors linked to both diet and disease that can distort results. An example, a plant-based eater may also exercise more, smoke less, and have better access to health care, not because of plants per se, but because of the lifestyle package. Even with statistical adjustments, unmeasured confounding often remains. So, once again, from a carnivore ketogenic implication, if a low carb meat eater were studied separately from a high carb meat eaters, many red meat is bad associations would weaken or completely disappear. Let's talk about recall and reporting bias. And this is the big one. Most nutrition epidemiology relies on food frequency questionnaires or self-reported dietary intake, right? With diaries. People misremember what they ate all the time. Many underreport unhealthy foods and overreport healthy foods, right? Uh, uh, People just do that naturally. So, from a carnivore and ketogenic implication, someone who's metabolically healthy on a high meat, low carb diet may report less accurately because the questionnaire isn't designed for their eating style, often lumping steak and processed meat together. Okay, small effect sizes. This is a big one. Relative risk increases of 10 to 30% are often reported, but anything under 100% increase, right? A relative risk that's less than 2.0 in observational epidemiology, that's a statistical way of rating this, right? Is generally considered weak and potentially spurious, false, right? Many nutrition studies report relative risks of 1.1 to 1.3, which can vanish when better data or randomized trials are done. A classic example of this is statin use, right? The statin drugs that you were told you're supposed to take all showed relative risk increases of 10 to 30%, which, when you looked at the actual real data, represented basically 3 people getting heart attacks out of 10,000 instead of 1 person getting a heart attack out of 10,000 hours over 10 years. Right? It was so incredibly small. It was like it didn't apply. Okay? So, from a carnivore ketogenic implication, these small risk signals are often used to demonize nutrient dense animal foods despite the effect being within statistical noise. You got to look at the studies and say, what are they studying, how are they studying it, and what is the real risk? Relative Anything that's reported in a relative risk and it's not at least 100% or better relative risk is usually not worth even worrying about. Okay, healthy user bias, and this went into like the reporting. People who follow mainstream healthy eating advice, low fat, high fiber, more plants, tend to exercise more, avoid smoking, have higher incomes and education. This inflates the apparent benefit of these diets in observational data. Conversely, people who ignore official advice and eat more meat are more likely to have other health factor risks unrelated to the meat. Though, I'm going to tell you right now, that part's going away, right? People who self identify as eating ketogenic and carnivore need to be taken completely out of the people who eat more meat bias because they are very healthy compared to people who eat more meat, but who meat is eating, and this is from one statistical study, eating a pepperoni peanut was considered eating meat. Yeah, that's real healthy. Okay, reverse causation. Sometimes people change their diets because they are sick, not the other way around. For example, someone with heart disease may switch to a heart healthy diet, thinking that that's better for them, which could make that diet look worse in certain analysis. Okay? Or the other way around, it could just be that the diet is worse for you. Okay? You gotta be very careful with this. Alright? Alright. Low reproducibility. And this is very important. The same epidemiological data set can be used to prove opposite conclusions, depending upon which co-founders are adjusted for, how food categories are grouped, and which subpopulations are analyzed. And that's probably one of the biggest things people are now doing is they're going back and they're looking at some of these old epidemiological studies and they're looking at the data and they're analyzing and they're finding, oh, the conclusions were completely wrong based upon the data given, right? Epidemiological studies can lead to many different types of conclusions. Okay, so bottom line, from a carnivore and ketogenic perspective, nutrition epidemiology is useful for generating hypotheses, right? Conclusion, things that you can test, but not for making definitive dietary guidelines. Stronger evidence comes from randomized controlled trials, RCTs, mechanistic studies, and ancestral evolution and biology. And when you're talking about humans, RCTs are very hard to do because of the control required to do a true RCT and the cost, right? So, mechanistic studies, identifying how the gut is built, right? How your GI tract works, how the cells actually work, cellular biochemistry, much more important than dealing with RCTs. And ancestral evolutionary biology, right? Remember, I mean, we've got uh, good good evidence from 10, 20,000 years ago, and maybe a lot more on how healthy humans were and how things have changed, and we can look at that. Okay, when RCTs test ketogenic or carnivore diet styles, and this is important, we've got several studies now that use very rigorous RCT controls. Results often contradict epidemiological warnings, showing improved metabolic markers, reduced inflammation, and better weight control. So, the weight of the evidence really leans towards carnivore and ketogenic diets. The logical issue is treating weak association only evidence as proof, right? Which ones can? Most of the people tell you to eat non-carnivore diets to lead to where carnivore people who are advocating carnivore will advocate towards more strenuous science, right? So this leads to policy that demonizes red meat and animal fats without robust causal evidence. Okay? Let's talk about rodents, right? So the problem with rodents is that they have a completely different physiology and metabolism. So their digestive system, rodents are omnivorous granivores. They eat primarily seed and grain. With a gut and microbiome adapted to high fiber plant matter, humans are facultative carnivores with smaller colons, larger small intestines, and different digestive enzyme profiles. Metabolic rate in rodents is much higher resting than the with a much higher resting metabolic rate and body temperature than humans, which can affect how nutrients and toxins are processed. And rodents have a completely different fat handling mechanism. Rodents metabolize fats and cholesterol very differently. Many strains naturally develop high cholesterol in diets that wouldn't affect humans the same way. So you can't use rodent studies on cholesterol to determine anything in humans. So the implications: a diet high in saturated fat might spike cholesterol in rodents, but in humans on a carb restricted diet, LDL particle size often shifts to a less atherogenic pattern, which is healthy, and triglycerides drop dramatically, which is also healthy. Okay, species specific nutrient requirements. Rodents require nutrients humans don't. For example, vitamin C synthesis is not, humans just can't do that, but rodents can, right? Some compounds metabolized into harmful byproducts in rodents are harmless in humans, and vice versa. And protein sources have different effects. Casein, soy, and meat protein, which, and you see a lot of rodent studies with casein, for example, can produce different hormonal and lipid responses between the species. The implication: a toxic effect seen from red meat in rats may be irrelevant in humans due to different amino acid handling and gut fermentation patterns. Okay, artificial diets in lab conditions. So, rodent chow is highly processed, shelf-stable mixture, often including purified sugars, isolated protein, seed oils, and synthetic vitamins. And there's a reason for that. when you're trying to do a nutritional study, you want to get everything reduced down to as minimal variables as possible so that you can change one thing and see what that one thing does. Which you can do with rats in a cage. You can't do that with humans. Okay? It just doesn't work. High fat diet in rodent studies is usually 45 to 60% calories from fat plus refined sugar and soybean oil, not ribeye steaks or whole food keto fats. You're checking the wrong kind of fat, usually. And animals live in confined, stress free, or what we might think is stress free, or very stress inducing. It depends on how crowded the cages are. Lab settings, far removed from human lifestyle complexity. Okay? So the implications. Results often tell us what happens when the rodent is fed junk, fat, plus sugar, not what happens when the human eats nutrient-dense, whole animal foods. Genetic homogeneity, and this is very important. In lab rodents, they are bred to be genetically similar, which reduces variability but doesn't reflect human genetic diversity. Humans have vast differences in metabolic flexibility, fat tolerance, and insulin sensitivity. So you can't trust a rat study to be valid against humans because of that. Okay, you just really can't. And a lot of the rat studies that we see and I've looked at and commented on, they use genetically malformed rats. They induce a genetic problem in the rat that accentuates what the study is looking for, and all the rats, of course, are then bred to have that abnormality. Well, that's not normal. Okay. Okay, short lifespan and accelerated disease models. The average rodent lives 2 to 3 years, right? Disease progression is compressed and may follow different pathways than humans. And you all heard the stories, right? You feed the rat a high dose of something, it gets disease. Something that you'd never actually eat that much of that product, right? Cancer, diabetes, and heart disease models are often induced artificially with toxins or extreme diets not naturally developed as in humans. So the implication, a rodent cure or harm might not apply at all to humans. They only apply in the specific engineered disease state. Historical mistranslations. Oh, this is a big one. Many compounds that look harmful in rodents proved harmless in humans. For example, remember this whole big thing, saccharin bladder cancer scare? Saccharin's not good for you. Okay, I'll tell you that right now for many mechanistic reasons, but not that it caused bladder cancer in humans. Okay, others that look beneficial in rodents failed in human trials. Right? Yeah, rodents love certain stuff and it's good, healthy for them, but it's not healthy for humans. Okay, so bottom line, right? Rodent studies can suggest mechanisms and genetic hypotheses that you can hopefully test later on in other ways, but they cannot determine optimal human diets. They can human RCTs and ancestral diet evidence and metabolic health markers are far more reliable, and these are the things that we use in the carnivore keto sciences. And when tested in humans, high quality ketogenic and carnivore diets often yield metabolic improvements that rodent high fat studies would have predicted to be harmful, right? Because you're not a rat. Just keep that in mind. So stop worrying about the danger of direct extrapolation of rodent studies against humans. It just doesn't work, okay? All right. Thanks for watching. If you have any comments or questions, put it in the appropriate section. This is a community we're building here. I'm trying to help provide you the answers that you want to have to questions that you're asking. Let me know what you're looking for. We'll put it in there. Thank you so very much for that. I can only grow by answering your questions, right? And as always, if you haven't done so, please like this video, subscribe to the channel, share it with a friend. It helps the channel a great deal, and you'll be helping a lot of other people, and I do appreciate that a whole lot. Thank you very much. Have a great day.